Assalamu alaikum. So this is the last part of the enzyme regulation lectures. Um, in this lecture, what we will do is talk about the last mechanisms of regulation that include regulation of enzyme amount, location, and non-specific regulation as well. And then we'll talk about the use of enzymes in diagnosis, disease diagnosis, and modes of regulation at the end as well. So let's talk about regulation of enzyme amount. Now the thing is, a cell should control how much enzyme it has inside. So what it can do is to regulate the rate of synthesis of enzymes, and this can be done at the gene level. Okay, so an example is we talked about steroids in the molecular biology course. Now, one thing about steroids is that they can regulate transcription or regulation of gene expression at the transcriptional level. So what they do is that they get into cells, since they are lipophilic. Okay, they get into cells, they bind to the receptor, and the receptor, this complex, is really a transcription factor. So what it does is that it binds to the promoter region of certain genes that encode metabolic enzymes. Okay, so you have induction of transcription. Now this is actually a mechanism that is used by androgens. So androgens are responsible for, or class of genes that is regulated by androgens is those involved in lipid metabolism. Okay, cortisol is another hormone that regulates metabolism. This is why people who take cortisol, they feel that they are hot. They have hot flashes. They feel energetic to a certain extent because of increased metabolism. Okay, now at the same time, the level of enzymes can be regulated at the level of stability. So what happens is that some enzymes, enzymes can be degraded just like other proteins, and they're degraded by proteases. Now, these are some examples. So you have catalase. It has a half-life of 1.4 days. Okay, so almost a day and a half. Now, some enzymes, by the way, have a half-life of hours. So it really varies among different enzymes. Glucokinase, it's about a day or so. Okay. If you look at lactate dehydrogenase, the different isozymes, not only that they have different half-lives, like the LDH1 in the heart has a half-life of about a day and a half. If you look at the LDH5 in liver, it has a half-life of 16 days. Okay, so that's a huge difference. Now, not only that it is isozyme dependent, it is also tissue dependent. So if you look at LDH5, it has a half-life of 16 days in liver versus 31 in muscles. This is a huge difference. Now, this is not really for you to memorize. Okay, I just want to show you the differences in half-lives of different enzymes. So now something else is that tissues can regulate synthesis of isozymes. So for example, we talked about LDH1, right? And I said that it is predominantly expressed in the heart. Well, the heart muscle also expresses LDH2, by the way. Okay, so now there are other examples of cells or tissues switching one isozyme versus the other depending on the condition that this tissue is exposed to. Okay, so under stress conditions, for example, you have tissues favoring the expression of one isozyme versus the other. Okay, so keep that in mind. The other level of regulation is compartmentalization. What we mean by that is by placing enzymes in certain compartments inside cells. okay, or like vesicles, for example, placing hydrolytic enzymes in the lysosomes. Now, the idea is that, remember that enzymes are not really that smart. So it's not that when they see a substrate, they run towards this substrate and they try to catalyze the reaction. It's not like that. It's all dependent on collision, random collision. And the probability of collision increases if we place enzymes and substrates in a small compartment. So that increases the rate of collision. Okay, so by placing hydrolytic enzymes and proteases inside lysosomes, all we have to do is just take the substrate from outside or inside the cell, and we place it in lysosomes. So now this is really a small area by which we increase rate of collision, and as a result, rate of reaction. Now, the same is true in other compartments like the nucleus, ER, and so on. Even the membrane, the plasma membrane, you have enzymes placed in the plasma membrane. So instead of enzymes and substrates moving in three-dimensional area like in the cytoplasm, rather they only move in two-dimensionally. Okay, so that increases the rate of collision as well. Okay, and we'll talk about this in the visual transduction lecture in your third year, inshallah. Now, the other thing is that this is sort of like a regulation of metabolism as well. So what cells do is that they divide metabolism into different compartments. So this is a smart way of controlling rate of metabolism by placing enzymes by placing enzymes of opposite function in different compartments. So for example, fatty acid synthesis takes place in the cytosol. However, fatty acid oxidation or degradation takes place inside the mitochondria. So this is a way by favoring or driving fatty acid synthesis or fatty acid degradation. Now, by placing them in different in the same compartment, that would require a different level of regulation. Okay, so what cells do is that they can take fatty acids into mitochondria so that they can be degraded, or small fatty acids can be placed in the cytoplasm where they can be elongated. Okay, they become larger. This is another mechanism by which the rate of diffusion can also be limited or restricted. Okay, so the whole idea is that, again, enzymes are not smart, and it's not like they see a substrate and they run towards this substrate, okay, and then they catalyze a reaction. Rather, it's all by random collision. So one way is by putting enzymes and substrates in a small compartment. Now, the other mechanism is enzyme complexing. Now, let's say that you have sequential reactions. So you have A converts to B, and this reaction is driven by enzyme 1. Then enzyme 2 catalyzes the conversion of B to C, and then you have enzyme 3 converting C to D. Okay, so you have sequential reactions. Now, the idea here is that if these three enzymes are separate from each other, you have the first enzyme colliding randomly with substrate A, okay, and then B is released. Now B would swim until it randomly collides with the second enzyme, okay, so that takes time. And then you have the third reaction after the release of C. Again, it collides randomly with the third enzyme and it gets converted to the final product, which is D. So instead of having these three enzymes separate, they are combined with each other. So they form a complex. So instead of B, okay, being released after its formation, it jumps or it gets transferred to the enzyme, the second enzyme that is attached to enzyme 2. Okay, so it doesn't leave the complex. Okay, and then you have C is, instead of C being released after its formation, rather it jumps to the third enzyme. Okay, so that limits the rate of diffusion, and it increases the rate of reaction. So a great example is pyruvate dehydrogenase. Now pyruvate dehydrogenase converts pyruvate to acetyl coenzyme. So that's pyruvate. It gets converted to acetyl coenzyme A plus CO2. So CO2 is released. Okay, this is part of the aerobic metabolism. Now this pyruvate dehydrogenase is really not a single enzyme. Rather, it's three enzymes combined with each other. Okay, now and the complex is huge. It is really large. It's composed of 60 polypeptide chains. Each one of them forms an enzyme. Okay, so you have multiple enzyme 1, you have multiple subunits of enzyme 2, and you have multiple subunits of enzyme 3. Okay, and that's how it looks like in reality. This is an electron microscopic image of pyruvate dehydrogenase. So that's how it looks like. So you can see the first reaction now is a decarboxylation reaction. Okay, removal of CO2 from pyruvate. Now you have inside this complex the three reactions taking place that include also oxidation as well as transfer of acyl group to the acetate or to coenzyme. Okay, we'll talk about this coenzyme in the cofactors lecture. So you have all of these reactions taking place all in one place in one complex. Now let's talk about non-specific regulation. Okay, one way is temperature. Temperature affects enzymes, of course. Now the thing is, the optimal temperature for human enzymes is not really 37, it's rather 40. Why? Because with the increase, with higher temperature, you have increased kinetic energy. So you have movement of substrates, faster movement of substrates and enzymes. So the rate of collision, the probability that enzymes and substrates would find each other is now higher. So that's at 40 degrees. Well, why is it that we see a decline? That is, here you have different temperatures, here you have increasing enzymatic activity. Why do we see a decline? Well, kinetic energy increases, yes, but protein denaturation takes place. So proteins cannot maintain their tertiary structure at higher temperatures. Of course, temperatures or the optimal temperatures for enzymes is different from one organism to the other. So remember in the molecular biology course we talked about Taq polymerase. This is the enzyme that is used in PCR to amplify DNA. And in PCR we use a really high temperature, right? So we denature DNA by increasing the temperature to about 95 degrees. Okay, and primers anneal at, let's say, 60 or so, and then you have DNA synthesis taking place at 72 degrees. Now, with the enzyme that we use to synthesize DNA is known as the Taq polymerase, which was isolated from bacteria or thermophilic bacteria that survive or that live at high temperatures. Okay, in hot springs, if you remember that. So the optimal temperature of enzymes is different depending on the enzyme and the organism as well. Now, something else that affects enzyme activity is pH. Now, what happens is that pH can alter the protonation state of the substrate and or the enzyme, right? So you have histidine, for example, needs to be in the protonated state. It should be positively charged in order for the enzyme to be active. Metals can also be oxidized differently at different pH values. Now, this is actually dependent on the enzyme itself. So you look at chymotrypsin, for example. This is a digestive enzyme that is present in the intestines. Now, the pH in the intestines is about 8, and the optimal pH of chymotrypsin is 8. Well, if you look at cholinesterase, we talked about this enzyme in the inhibitor lecture, the irreversible inhibition of enzymes. So cholinesterase has an optimal temperature of about 7 or so and higher. But when the pH is lower, when it is acidic, the enzyme is not active anymore. If you look at pepsin, pepsin is the stomach enzyme that is responsible for digestion in the stomach. Optimal pH is about 2. So that's the environment in the stomach. If you look at papain, papain is another protease that is isolated from papaya. Papaya is a fruit. Now, it's not affected by pH. So again, it is dependent on enzymes. Now, temperature is dependent on the organism, okay? So, these are different ways by which enzymes are regulated. So let's talk about modes of regulation, different types of regulation, especially we're talking about metabolic pathways, okay? So in metabolism, you have sequential reactions. So you have A is converted to B, B to C, to D, to E and F, and each reaction is catalyzed by a specific enzyme. Now there are enzymes that are really important. They have to be regulated in order to accelerate or to slow down metabolism or to block it altogether. Now, one way by which metabolism is regulated is what is known as feedback inhibition, meaning that when you have the production of F and the level of F is really, really high, there's no need to convert A to B or to drive this reaction. So what happens is that once you have this final product or any one of these products formed at high concentration, you have this product coming back and blocking the initial reaction or one of the previous reactions. And a great example is glycolysis. So in glycolysis, you have glucose converted to glucose 6-phosphate. Well, the thing is, by hexokinase. So the thing is, if the level of glucose 6-phosphate is really high, there's no need to phosphorylate more glucose. So you have glucose 6-phosphate inhibiting the activity of hexokinase. So this is known as negative feedback or feedback inhibition. And this is known as product inhibition as well, since the product inhibits the enzyme that produces it. Now, another mode of regulation is feedback activation, meaning that you have the final product actually activating the initial reaction so that more of F would be produced. So this is feedback activation, or it's also known as positive feedback regulation. Now the idea here, or a great example, is blood clotting. If there is injury and one of the reactions, one of the final reactions in blood clotting is the formation of thrombin, which is a protease. Now when thrombin is produced, this is a signal to the body, to the enzymes, that there is injury that must be blocked, that must be closed, that must be treated. Clotting is important to stop bleeding. So thrombin accelerates the reaction that produces it. Okay, so you have more thrombin produced in order to induce blood clotting. Okay, and we will talk about these reactions later on in the hematopoietic system, inshallah. Now, you have also feed-forward activation. That is one of the initial reactions, one of the initial products accelerates a final reaction by interacting with the enzyme that catalyzes this final reaction. Okay, so an example is poisoning, for example. So if you have A introduced into the body and A is a poison, now A converts to B, then to C, then to D. Now, what happens is that A can induce the reaction that converts it to B. It's a signal to the body that A is a poison, and the removal of A must be accelerated. So you have one of these reactions actually activated by the initial substrate. Okay, a great example of what is glycolysis, the breakdown of glucose. So you have glucose to glucose 6-phosphate to fructose 6-phosphate, etc. And you have the final reaction that is pyruvate. This reaction is catalyzed by pyruvate kinase. Now this pyruvate kinase is induced further by fructose 1,6-bisphosphate. So the existence of fructose 1,6-bisphosphate indicates to the cell that energy is needed. Accelerate the reaction, the final reaction, so that energy is produced. Okay, so this is feed forward activation, which is part of feed forward regulation. Now, in these reactions, there is something known as a committed step, which is basically a reaction that means there is no point of return. That's it. The pathway must continue till the end. Okay, so usually these committed steps are exergonic. Okay, so going back is not a good idea, because going back would mean driving an endergonic reaction, which is unfavorable. Okay, it's not spontaneous. So for example, you have A converting to B. Now B has two fates. It can either be converted to C and then to D and then to E, or it can be converted to X, then Y, then Z. Okay, now, so if I ask you, does the formation of B mean that B would be converted to Z? You would say no, because it can be converted to C. So this is not a committed step. It doesn't mean that this reaction is driven, is catalyzed, that B would be converted to either Z or E. Okay, so there's no guarantee. If I tell you, if B converts to C, does that mean that C would continue all the way to E? You would say yes. Okay, because once C is formed, there's no way back. That reaction is not reversible. Okay, so the committed step for the formation of E from A is actually BC. That's the reaction. It's not the first one. So if I ask you, does the formation of X mean that B is committed to be converted to Z? And the answer is no, because C can be converted back to B, right? Because this reaction is reversible. So what is the committed step? The committed step would be the conversion of X to Y, because Y has to continue to the formation of Z. So this is the meaning of a committed step. Now, so let's look at again glycolysis. So you have these 10 reactions: glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, all the way to pyruvate. If I tell you what is the committed step in glycolysis, and if you say, oh wait, it's glucose to glucose 6-phosphate, okay, I would say why? And you would say, because this is an irreversible reaction. I say, oh, that's great. Now, does that mean, is it guaranteed that glucose 6-phosphate would be converted to pyruvate? And the answer is no, because once you have glucose 6-phosphate, it has different fates. It can be converted to pyruvate via glycolysis, but at the same time, it can be converted to glucose, okay, or it can be converted to glycogen. It can be added to glycogen, or it can be converted into 5 carbon sugars that in a pentose phosphate pathway. Okay, so that's not the committed step. Okay, this is not the committed step. So what is the committed step? Is it glucose 6-phosphate to fructose 6-phosphate? And the answer again is no. Why? Because it is reversible. Okay, now it's this reaction right here, fructose 6-phosphate to fructose 1,6-bisphosphate. Why? Because once you have fructose 1,6-bisphosphate, that's it. There's no point of return. It has to continue all the way to the formation of pyruvate. So that's the committed step. Okay. Something else about reactions in metabolism is that some reactions are rate limiting. What do we mean by that? Let's say that A converts to B, B to C, C to D, and then to E. Now, if you look at this reaction, you have this reaction is fast, okay, and the rate of reaction is really fast. Now, if you look at the second reaction, B to C, it's fast as well, okay. But if you look at the reaction of C to D, now this is a slow reaction. Okay, so this is rate limiting. This is the bottleneck. This is the reaction that is really slow. Now, why is it slow? Because either it requires high amount of energy. So remember that cells are cheap. Okay, they do not like to spend energy. They are lazy. They'd rather spend energy for useful things. Okay, so any reaction that requires energy is really slow, and it is rate limiting. So if you look at the first reaction right here in glycolysis, glucose to glucose 6-phosphate, it requires energy. Okay, so this reaction is really slow. If you look at the second, if you look at this reaction, fructose 6-phosphate to fructose 1,6-bisphosphate, it also requires energy. So it is also a slow reaction. Now, the third reaction as well, the final reaction as well, is slow and is rate limiting. Now, the other thing is that you have a reason why some reactions are rate limiting is that enzymes that catalyze these reactions are highly regulated. So the first reaction by hexokinase is really highly regulated by many factors, including glucose 6-phosphate, ATP, and so on. So if there are high levels of ATP, now ATP would actually inhibit this enzyme. Same thing with the enzyme phosphofructokinase 1. It's also highly regulated. But all of the other reactions which are reversible, they are not really regulated, so they are not rate limiting. Now, another reason is that enzymes have low affinity towards substrates. They have high Km values, meaning that even if they bind to a substrate, there's a good probability that this substrate would be released. Now, usually rate limiting reactions are also committed steps. So let's talk about enzymes and the use of enzymes in disease diagnosis. Now, the idea here is that enzymes are present inside cells to a large extent. Of course, you can have enzymes in serum, in blood, but in general, enzymes are present. Certain enzymes are intracellular inside cells. Now, when these enzymes are present in blood or serum, that would indicate that this cell or this tissue is damaged. Okay. Now, by the way, cell death and tissue damage occurs all the time. So it's really normal to have an intracellular enzyme in blood. This is fine. Cells die all the time. But if there is tissue damage, there would be extensive release of enzymes into the serum. So their level would be much higher than normal. Now, examples include aminotransferases like alanine transaminase, aspartic transaminase, and so on. Another example is lactate dehydrogenase. A third is creatine kinase or phosphokinase. Okay, so let's talk about these enzymes now, and let's talk about enzymes that are used in diagnosis. Now you have aspartate transaminase and alanine transaminase. Now these enzymes are mainly liver enzymes, okay? However, alanine transaminase is predominantly present in hepatocytes, in the liver. So if you have high level of alanine transaminase compared to aspartate transaminase, this can be diagnostic. It can be diagnostic of liver disease. If you have high level of alanine transaminase compared to aspartate transaminase, it means that there is viral hepatitis. There is viral infection. Now, if the level of aspartate transaminase is higher than alanine transaminase, it can indicate liver disease or damage that is not of viral origin. Now, you have multiple proteins and enzymes that are biomarkers of myocardial infarction, but some of them are really not that specific. For example, let's say that you have an onset of myocardial infarction. That would result in tissue damage, extensive tissue damage, and release of many proteins and enzymes that are present in heart muscle cells. Okay, that would include myoglobin. So after the onset of myocardial infarction, within minutes you have release of large amount of myoglobin into the blood. But myoglobin is not indicative of myocardial infarction. Why? Because it is not really a specific biomarker. That is, let's say that someone has chest pain. Someone has, chest pain. Now, this person freaks out and this person runs to the emergency room. When he gets to the emergency room, there will be high level of myoglobin in this person's blood. Why? Because one source of myoglobin is muscle. So during this running, you can have damage of skeletal muscle and the release of myoglobin. So myoglobin is not an indicator of myocardial infarction. But rather you have other enzymes that can be indicative, they can be used as biomarkers of myocardial infarction. So let's talk about these proteins. One of them is lactate dehydrogenase. But I have to say before going on is that lactate dehydrogenase is not really a biomarker that is used nowadays in diagnosing myocardial infarction. But let's talk about lactate dehydrogenase just to give you an idea of how things work. Now the thing is, normally, and if you look at the different isoenzymes of lactate dehydrogenase that are present in blood, each peak represents the amount of such enzyme. So you notice that the ratio of lactate dehydrogenase 1 to lactate is, well, you have lower amount of lactate dehydrogenase 1 than lactate dehydrogenase 2. That's what normally happens. And by the way, both of these enzymes are expressed by heart muscle cells. Okay, but heart muscle cells express more of lactate dehydrogenase 1 than lactate dehydrogenase 2. Anyhow, so normally you would have in serum more of lactate dehydrogenase 2, which is mainly expressed in red blood cells, than lactate dehydrogenase 1. But let's say that someone has myocardial infarction. This would indicate that you would have release of lactate dehydrogenase 1 from from heart muscle cells, right? So the ratio now is flipped. You have more of lactate dehydrogenase 1 than lactate dehydrogenase 2. So this is indicative again of myocardial infarction or damage to heart muscle cells. Now, but lactate dehydrogenase is not really a good biomarker for many reasons. But a good biomarker is creatine phosphokinase. Now, creatine phosphokinase exists as 3 isoenzymes: CPK1, CPK2, and CPK3. They are also designated as CPKBB or MB or MM. Now, the thing is, if you look at the level of, of these enzymes, these isoenzymes in serum, there would be a lot of MM compared to MB and BB. Okay? Now, if you look at skeletal muscle, they have a lot of MM, predominantly MM, very little MB and no BB. Now, if you look at the brain, there's a lot of BB, CPK-BB compared to MB and no MM. Well, let's look at cardiac muscle in here. You have a lot of MM and also significant amount of MB. Now, this means that if there is damage in cardiac muscle, you would have release of a lot of MM, but that means nothing because this can indicate damage in skeletal muscle as well, but you would have release of a lot of the CPK-MB isoenzyme. So this is really a marker of cardiac muscle damage. Okay? So, if you look at this plot again and look at total creatine phosphokinase, now less than 24 hours, you have release of a lot of creatine phosphokinase. But we are more concerned with the isoenzyme that is specific for cardiac tissue. And if you look again, if you look at this biomarker, you have a peak in less than 20, well, you have a peak at about 24 hours. So this is really a great indicator of myocardial infarction. Now the thing is, notice that it goes down quickly. Within 3 days, it goes down to normal level. But what stays up is what? It's lactate dehydrogenase, right? So let's say that someone comes in and says that he felt chest pain like a week ago. Okay, what can be done theoretically is that instead of looking at instead of looking at creatine phosphokinase or its isoenzyme, the MB isoenzyme, if you look at lactate dehydrogenase, you would find that it's present at high levels, right? Because it stays for a long time in blood. But CPK MB or total CPK cannot be used as a biomarker because it goes down after 3 days. So let's say that someone feels chest pain and this person comes to the emergency room and says that he felt chest pain 3 days ago and he feels with chest pain again at that time. So now looking at CPK-MB is a great idea because if there is a second incidence of myocardial infarction, that is reinfarction, you would have this CPK-MB going up as well. Okay, as a result of this second incidence of infarction. So this is so CPK-MB can be used as a biomarker for reinfarction. Now, look at this. Here we're looking at the levels of CPK isoenzymes BB, MB, and MM, as well as levels of the 5 isoenzymes of lactate dehydrogenase at different conditions. So normally, okay, let's look at lactate dehydrogenase now. In normal levels, there should be higher amount of lactate dehydrogenase 2 than lactate dehydrogenase 1, right? I mean, that's what we said. Normally, this would be higher than that. Well, let's say that you have myocardial infarction. Okay, so this is a person hours post myocardial infarction. You look at it and you say, oh, I cannot really tell because like they're almost equal. Maybe there's more lactate dehydrogenase 2 than 1. Let's wait till next day, or let's wait for a few more hours. So if you look at it now after a day, what do you see? You see that you have lactate dehydrogenase 1 more than lactate dehydrogenase 2, right? So you say, aha, that's myocardial infarction. Well, let's look at it after another day, and again, you see that the ratio is flipped. You have more of lactate dehydrogenase 1 than 2. Okay, let's say that someone comes in with liver disease. Now look at it, lactate dehydrogenase 5 is much higher than all of the rest, when normally there should be very little of lactate dehydrogenase 1 in serum, right? Okay, let's look at someone having liver disease and myocardial infarction as well. What do you see? You see the ratio of lactate dehydrogenase 1 higher than lactate dehydrogenase 2. The ratio is flipped, and you see very high levels of lactate dehydrogenase 5. So you can diagnose this person as having both myocardial infarction and liver disease as well. Now this here, the standard protein, is basically a sample that we know it contains all 5 isoenzymes as a way of comparing the electrophoretic migration or movement of the proteins in this gel. Okay, so we talked about electrophoresis before. So this is just to say, aha, this is actually LDH5. How do I know? Because it runs at the same speed as this protein right here, which I know is lactate dehydrogenase 1. And I know this is lactate dehydrogenase 2. Well, let's look at CPK. Now, normally there should be MM, but there shouldn't be MB or BB, right? Because having MB means that this person is having what? Is having tissue damage to cardiac muscle, right? There should be no BB at all, because that means that because it comes mainly from the brain, right? Well, let's look here. That's normal individual. These are standard proteins. It's a sample that contains all 3 isoenzymes so that I can compare the other bands to this standard, right? So I know that this is CPK-MM because it runs alongside the standard protein CPK-MM, and so on. So let's say that someone is having a myocardial infarction. Hours after this, after onset of MI, what do you see? Wow, you see a lot of CPK-MB, right? Normally it shouldn't be there, so that means that there is damage to heart muscle. Well, a day later and after 2 days, you can see the presence of CPK, but notice that it's going down, right? It's clearing up. Well, let's look at, so this is someone with MI again. You can see a lot of CPK MB as well. Now, how about someone with liver disease? Well, someone with liver disease, there should be a lot of CPK MM, but no CPK MB because it's not there. It's not in liver. That's someone with both CPK with MI and liver disease, and what do you see? You see a lot of CPK-MM coming from the liver, and you see a lot of CPK-MB coming from heart, right? So that's how diseases are diagnosed by looking at serum samples of patients and comparing them to normal levels. Okay, these are explanation of the different samples and what they represent. So, is CPK-MB used in clinical diagnosis of MI? Yes, but there is a much better protein known as troponin. So you remember that. Troponin is not an enzyme, it is an actin binding protein, and there are different isoforms of troponin. One of these isoforms is present in cardiac tissue, and it is specific for cardiac tissue, and it is an excellent marker that is used in clinical laboratory to diagnose myocardial infarction. And you will learn that later on in your clinical years, inshallah.